But with all that, thank you again, Alex. And now I'd like to introduce our other principal investigator of the IEDB, Dr. Bjoern Peters, to present an overview of the analysis resource and our tools more generally. Thanks very much, Bjoern.
Thank you, Nina, and thanks, Alex, for the database introduction. So as was just said, I'm going to talk about the tools, about the analysis resource in the IEDB. And what is kind of new, not kind of new, it's very new, is that we have now our first presentation of the real working next generation IEDB tool site. In parallel, this is a new implementation of the tools. And in parallel, we have the current tool site, which has more functionality. And I'll explain this in a bit. So starting with the next generation IEDB tools site, let me change here to a laser pointer. The next generation tool site is being launched this year, and it's been a multi-year project to get here. Essentially, our goals were to take the existing tools that were developed over 2 decades pretty much in a disparate fashion and try to really integrate them in a way that simplifies the user experience so that not every tool is a little bit different. Integrate them together to enhance the existing functionality, add new functionality, ensure consistency, that it's easy to switch from one to the other, and make the layout more pleasing to the eye. In this first release, we will only have tools for epitope prediction for class 1 T-cell epitopes. We'll have the cluster analysis and PEP match. So this is a small subset of the overview of the overall tools available. There's going to be much more information about this in day 2. In general, the presentation I'm giving right now is meant to give a very quick and rushed and purposefully rushed overview of the analysis resource so that the people can either hopefully find out if this is something they want to learn more about and then join again in day 2 for a lot more detailed introduction. So in these next generation tools, this is just a preview here of the homepage, where essentially our idea is that for each class of tools, you can just start with the most common inputs, such as for class 1 epitope predictions, a protein sequence and an allele, and then run that as an example from the homepage and get essentially an output directly that is then using the most frequently used user inputs. So in this case, for example, it would be for making predictions for 9-mer peptides for class 1. Then there's a still selection of prediction methods, which was then be just driven by our recommendations. But now in the second page, you can modify all these parameters and go into all kinds of details that you want, but there's defaults being set. Another key thing that is new that we're extremely proud of is the ability to build pipelines with this. So the tools can be chained together to implement workflows that you can then reuse over time. And this results table also has lots of functionality for downloading, sorting, etc. We also will have an integrated help site. Right now, again, like the tools are disparate and they are not really, you have to read the documentation for each one and they're slightly different. Here we're going to bring all these together and hopefully guide people better along. Now, as I said, in parallel, we still have, and we expect to have for at least another 2 years, the current epitope prediction analysis tool site. And that is our totality of tools we've ever made public. So we have T-cell and B-cell epitope prediction tools and the analysis tools. As for the next generation site, I'm going to rush through this here and just give you the flavor of what is going to be available. So when we talk about epitope prediction tools, those are really the ones where we have experimental data that was collected in the IEDB, and we are trying to generalize from that using machine learning models such as neural networks to extrapolate from the known data to predict new ones. This was, I mean, when we started, this was something we always had to explain, but now machine learning is in everyone's head already, so that makes it easier. So ultimately, on the training data, we are generating a model that we can then use for new input data and create new output data here. That's then the predictions. So The analysis resource can be accessed where these tools are by going directly to tools.iedb.org, or on the iedb.org webpage that Alex was showing. You have on the right here several links that guide you into the different elements of the analysis resource. This is the analysis resource homepage at tools.iedb.org, and these are essentially the direct access points to the tools, so the T-cell tools, B-cell tools, and analysis tools. In addition, what you have available here are access points to other modalities of running the tools, such as through an API or by downloading a standalone tool. We also make data sets available that were used for training the tools or evaluating the tools. And we have help in the form of the Solution Center, as well as tutorials, questions and answers. And you can always reach the IEDB team by emailing us directly. So starting with the T-cell epitope prediction tools, when you click there, you're getting an overview of these different kinds of tools that are available. One of the most frequently, and the frequently used ones are using MHC binding predictions, starting with MHC class 1 binding predictions. This is kind of what you end up with. You have your protein sequence input, you get to choose a prediction method, and we have a lot of different ones available here. And so we have here always the recommended line of which tool we would be recommending to run. And what is kind of new, not kind of new, which because there was always a bit of a confusion here, we have now separated out our prediction for if you wanted to actually predict epitopes, meaning peptides recognized by T cells after that are bound and presented by MHC class 1 molecules, versus if you really care about the binding affinity that you're getting, and then you have the recommended binding predictor. And the way we make these recommendations is by running benchmarks. This was the evaluation based on which we chose the T cell epitope recommended methods, where a large set of experimentally validated T cell epitopes in a model system were scored by all these different tools, and the net MHC pan EL or ligand elution one scored up top, be above the binding affinity predictions. We are also running like completely separately benchmarks on our newly released MHC1 binding data. You can see here each of these columns corresponds to a data set that was made available, and then you have the performance of different tools. And here what before for the binding affinity predictions, so really evaluating the ability to predict IC50 values, the net MHC pan BA methods, this is the binding affinity methods, performed the best, actually much better than the ligand elution methods, which makes sense. So again, like depending on what you want to do, you might want to choose one over the other. What is coming up right now is a new benchmark that we wanted to run, where we're going to evaluate the ability to directly predict ligand elution data. So just to be clear here, MHC binding measures the direct molecular interaction of a peptide and the MHC molecule. Ligand elution identifies what peptides can be found on the surface of MHC molecules, and T-cell epitopes are those that trigger an actual T-cell response. So the problem with the running these benchmarks here, what makes these non-trivial, is that we need some kind of negative data set and to distinguish positives from negatives. And there we have actually made found that using UniProt generated peptides gave us the best, most relevant data from in terms of negative data sets. So this is how these prediction methods were, these recommendations are made. Then you enter an allele and a length. You can add an email if this is going to be something that's going to take a long time to run. And then you get your MHC1 binding prediction results. There's always some guidance here, specifically in this case that the high score is a good binder. This can always be confusing. Some methods have the opposite. And this corresponds then also to a low percentile rank. So these are sorted by default so that the best predictions are up top. MHC2 binding predictions. very similar kind of input data that you have. There are differences, and again, it's going to be talked about a lot more tomorrow. We also have here our MHC binding prediction benchmarking, and here, the net MHC 2PAN 4.1 BA again performed the best in terms of the binding affinity predictions, and we have, similar to for the T cell ones, a recommendation for the EL method for T cell epitopes. So these are like the bare bones tools essentially that you can use here for the MHC1 or MHC2 binding predictions. And there's a lot of additional steps that are typically necessary starting from your sequence, like where you say stuff like, do you want to remove redundancies? Do you want to start off with like overlapping 15 mirrors spanning by 10? That's a common thing we do for class 2. All kinds of steps. And this TEPI tool implementation here uses the same backend for making the actual predictions, but guides you in a stepwise fashion through the prediction process and the typical pre and post processing steps that we have found that we typically run. So this would be our recommendation actually to start off here if you want to start making predictions and then trying to figure out how to get at the useful results. Yeah, and then this is how the output looks like in step 6, where you then similarly can get your put your email in and run it to yourself and get a notification when the results are done. So switching from T cell to B cell tools, here we have the tools available here. Is again on the tools.iedb.org tab. You have the B cell predictions listed here, and here the distinction is not so much class 1, class 2, that doesn't exist for B cells. It's the linear versus structural predictions. So in the linear epitopes predictions, you have again like an input sequence that you're giving. You can choose a number of methods. We would strongly recommend to use out of these the BepiPred 2.01, which performs by far the best in the benchmarks that we have been doing on this in the past. And essentially what this method does, and all the others as well, is run through the sequence and identify areas here, scoring one residue at a time, identifying areas that consistently score high for the likelihood to be in B cell epitopes. And then you have outputs like the overall graph that highlights this on the position in the sequence, the predicted peptides that are then listed individually, and the for each of the positions in there, the score that you get. And then what becomes a predicted peptide based on these inputs can be modified by looking by modulating the thresholds being used here. So antibodies bind not linear segments in in the protein, but they are actually can recognize the folded 3D structure of a protein and the discontinuous patch of residues on it. So in order to make predictions for that, you need to start off with an existing structure, which, if you're lucky, is available directly in the PDB based on experimentally generated data. Then you can just enter the PDB ID in there, the chain in the PDB ID, in the PDB structure. You often have multiple chains that are coming together to form your protein, and you need to identify which one is it that you care about. You can enter the version of Discotope, and you're getting an output like this, which now again here in this case displays it on the protein sequence, and you have like a similar output like before, but then you can take this and start the 3D viewer that actually displays the same data on the protein structure, and then you can see how some residues that are in the linear sequence far apart are actually close together in the 3D fold of the protein. So what I showed you so far were the running the tools directly on the web server. Alternatively, you can download actually the executables that make the ultimate predictions and have them on your own computer. The advantages of that is that you don't need internet access, obviously, at that moment. Most importantly, if you're running large data sets, this means you're not going to be bound by connectivity issues or transfer rates for the data sets or so. And also our server is going to be very, very slow for a significantly large data set, and it's going to be much faster for you to run it on a local machine. The other thing is actually confidentiality. Many companies that want to protect the sequences that they're running through our tools and are not allowed to use it outside of their firewalls wanted to have these things implemented. So we are making packages of these like virtual machines implementing the whole tools behind their own firewall available. And if you're interested in that for commercial use, please contact us at the license at IEDB.org. For academic users, this is all, including the downloads, etc., is all free. So we should be thankful actually for the commercial users that are paying for the license fee that actually funds the maintenance and support for these other modalities of access. So once you have this local, you can run this tool on your favorite Linux cluster or Linux local machine or so. You can take your input sequences here and the alleles that you want to make predictions for and generate these kind of output formats. And yeah, the other approach, this is all requires local installations, is to actually use our API. That's the way how you can talk directly to this web server from computer to computer without having to go through web forms. This is also freely available. And if you have like an existing pipeline that at some step wants to make a prediction, this is probably the preferred approach. It also benefits from anytime something gets updated, you don't have to reinstall anything. You can always like directly use the same code to use the most current prediction tools that we have. There's explanations in here on how to run the API. And here's just as an example, you might have a file like this with you have your different peptides and different alleles. That's actually quite annoying to input these kinds of things on the website. And then just run a script in which you have your API call integrated to make a prediction. And then you get your output in any way you want it to. And you can imagine, like if you're happy with coding, then this is going to be a very convenient way of integrating these predictions into your code. So to summarize these access modalities, we have the web page where you can go in with your regular browser, and obviously this should always be the starting point. You need internet. That's maybe a bit of a downside, but not really, I think, these days. And it's definitely not good for large data sets, but this, I think, should always be where you get started. If you want to run very large data sets, the standalone is going to be more efficient for you in terms of speed, definitely, and avoiding large data transfers. And the API is probably the most elegant. If you're very happy with coding and want to integrate this somewhere else, then this is the most efficient way of implementing that. So to summarize here, what are some points to remember? Something that I didn't actually mention, but I mean, Alex talked about the database. Before you make predictions, you should always check, has this been done already? Has somebody already studied this? Can I just utilize experimentally verified data rather than predicting something? That will always be preferable. There are as I said, like the prediction methods we have, the extrapolate from existing data by identifying patterns and then allowing to predict data for new sequences. We are using ROC curves, AUC values, as our preferred metric of performance. Also very importantly here, the prediction is a screening step. It's not confirmatory. So if you run a prediction and you get a peptide out, you can't say, oh, this is now an epitope. It is a candidate that you should experimentally test for verification. And if you do large-scale analysis, and you say like this is a list of candidates that are more likely, that's fine if you just do a large-scale computational analysis. But you cannot call something an epitope just because you have a prediction for it. The analysis tools, which I didn't talk about here, we're going to talk about later, are not so much about predicting new epitopes, but for existing sets of epitopes that you might get from queries or from predictions. And there's not really a performance metric for them. their performance here is the usefulness, and there's a broad area of applications for them, which again, we're going to go through tomorrow. That takes me back to my first slide. So we have, again, like the tools.iedb.org, which is most of what I talked about. And then we have the next-gen tools.iedb.org, where we have the new implementation, which is now ready for access. So test them out. That's the most fun part, to figure something out. Try, if what I'm talked about is interesting to you, go there. Nothing should stop you. And with that, I will stop. Look forward to questions.
Perfect. Thank you so much, Bjoern. Perfect timing. So we are going to move into our 15-minute Q&A. So at this point, I'll invite Alex back to join Bjoern. And I do just firstly want to say, while Bjorn's reviewing the Q&A, thank you to our attendees. There are already a ton of questions coming in, so we really appreciate your engagement already. And one thing I didn't mention is we will answer as many questions as we can live, but of course there are so many coming through that we try and be efficient and also respond via typing. So don't forget to check the answered column in the Q&A for any questions that have been answered with a typed response. But with that, Alex, I will hand it over to you to take care of the first question.
Yes, and I cheated because while Bjoern was talking, I answered most of the questions that were related to the database. I see a question, how many curators are working on this project? There is also a question maybe for Nina, but that has changed over time. Also, curators are not only curating, they do a lot of other stuff, including peer review, recurating things that need recurated for a variety of reasons, and also are involved in other research projects. Nina, I think that what's the current headcount for curators about?
Great question. I did double check. So we currently have a team of 10 curators, so 6 full-time curators, 4 part-time curators. And in addition, we have 1 document specialist that helps facilitate the whole curation process, as mentioned, curation, peer review, and other tasks.
And also all the full-time curators are not only curating, so they are full-time employed, but they wear multiple hats. Then I don't know if there's a question, how can you filter search to epitope tested in lateral flow assays? So there are different category of assays, and I believe Randy will get more into detail of the different assays and how they're categorized. So hold on for that. Bjoern, do you want to start answering some questions?
So Alex, if you can use this answer live thing that you want to click on for the ones that you want to answer, and then I'm doing that for the ones that I have. So, okay, so I had a question from Akbar, which says, it would be nice to show the name of the sequence for these predictions rather than the sequence number. So we had that before. There's actually at the top of the predictions, there's the name of the sequence, like the protein sequence. And that's like that is numbered. So you can kind of look up number 3 is whatever the hemaggluteinase, whatever. The reason why we don't repeat it is it becomes really large. And then the amount of text on your screen and the amount of data that you download is largely going to be repeat of that same name over and over. So that's why we have that lookup. And if I do a second one as well, Sham28 asks, is the updated analysis tool also available for MHC2? I mean, the epitope prediction, not the binding affinity only. So yes, so the net MHC pan net MHC2 pan 4.1 EL method is the one that predicts not just binding affinity, but predicts likelihood of processing and presentation, which we have found to be better when you want to predict epitopes versus the BA method is best in predicting quantitative affinities. Alex.
Yes, there's a question. There were a number of questions relating innate immunity and PAMP receptors and things like that. And I just want to make to clarify that the IDB scope, what is our mandate from NIAID, and is to curate adaptive immune epitopes. So epitopes interact with MHC or T cell receptor or B cell receptors. So we do not curate innate immunity epitopes, not because they're not important, not interesting, but also the resources are limited and we need to focus our efforts in particular fields. There's another question that was, can the IEDB validate or propose experimental assays to make sure that the predicted epitopes are really epitopes and so forth? And the answer is no, in the sense that the IEDB is not generating any wet lab data. So certainly the validation of predictions by experimental work is a very, very important activity, and Bjoern alluded to that, but this is not something we do or we endorse one particular method or another. It's, again, not within the scope of the database, which is to just, as objectively as possible, report what people have done. And you can think about it like when you do a search for PubMed for a particular search term, you don't want to find the papers that the PubMed personnel thought they were good. You just want to get everything that was published related to that field and then go from there. Bjoern.
Okay, I'll take the next one. So Manikanta Bondada, and I apologize for butchering names here, asks, is NetMHC Pan 4.1 BA versus EL, which is best for finding epitopes? And there is the EL method that we would recommend in terms of prioritization, because it doesn't just incorporate the binding affinity predicted for a peptide, but also includes the kind of motifs that make it more likely to be processed and presented by things like proteasomal cleavage and tap transport. The next question is from Lorna Kelly, now somebody I know. Do or will CEDAR and the IEDB communicate with each other such that if I query a neoantigen epitope in the IEDB, will it also query CEDAR for an output? So yes, actually the database that is being used is the same data that in the case of CEDAR is being queried in a kind of like with a cancer focus. But so say if somebody measures the binding affinity of a peptide that is known to contain a cancer mutation, that would be popping up in both the IEDB and CEDAR. In CEDAR, it'll be easier to query for things like, I want to only query for mutated peptides. That, for example, in the IEDB is not a primary focus. So the database is the same, but the kind of like focus of the user and accessibility of the data has a different focus because these are different user community. But yes, there's going to be immediate benefit one way and the other, specifically for cases where it's like not even clear. It's like papilloma viruses. Is that an infectious disease or cancer? I mean, it's both. So there's always a scope for, there's usefulness for this. So Alex, go ahead.
Sorry, got confused. What was the, so there was a question, can you use a standalone on a university supercomputer? I don't see why not. Then there was another question related, can the tools be used to predict glycopeptides or other post-translational modifications? And in general, that is a useful thing, but it's not currently hosted. The main issue is that in order to do this kind of predictions, you need data to train on, and that has been not abundant, and certainly also it stands to reason that the effect of a post-translational modification may be different depending on what is the specific post-translational modification. So it's a topic of interest. We actually have a research interest also with collaborators, and something may be developed in the future, but currently is not available. Is that correct, Bjoern?
Yes. Okay, I have next a question from an anonymous attendee. BepiPred 3.0 has been released. It's quite different than 2.0. They use PLMs in the latest version. Do you consider to deploy BepiPrep 3.0? I would actually love if Jay can later maybe put it in the chat, but my recollection that there was a problem with BepiPrep 3.0 because it was relying on, as part of it, on code that we do not have the ability to host on our server because of licensing issues. Otherwise, we will always update to the newest versions and have these suites of tools there. So Jay or someone else, if they can put for the answered question, I think maybe add to it if that was wrong. Then the next question is from Hur Al-Hassani. Why the recommended tool reports score while calling the tool directly reports IC50? So the examples I showed were for the EL methods, where the score can be interpreted as like a probability of it being an eluded ligand. So roughly, many of the methods have some kind of score, and in the case of the BA method, that can be directly translated to an IC50 value, yes, but others don't. In the complete output version, you will have access to both the IC, if there is an IC50 output, that will also be reported. Alex.
So Manikanta Bondada asked, what is the difference between epitope analysis and next generation tools? So in general, the epitope analysis tools are tools that, as we will be discussing later on, that relate to analyzing data that you might already have. So I have this series of different sequences. Can I cluster them in some way because they're largely overlapping? Or I have this series of epitopes. Are they conserved in a set of different influenza virus sequences and so forth? So you're not predicting, you're just analyzing. While the next generation tools are in general part of a category of predictive tools, where you have a sequence and you want to predict whether there are B-cell epitopes or T-cell epitopes or eluded ligands and so forth. There was another question. Is too strong BA value means it may be anergic? And also explain how low versus high BA value in terms of protection and long-term memory response, if any. So this is a general, excuse me, a general issue relating binding affinity of epitopes with their biological activity. And there is no binding affinity that makes something tolerogenic or immunogenic or induce good memory versus non-durable responses per se. In fact, the same epitope with a binding affinity in different contexts may be tolerogenic or generating strong responses. There's been questions, for example, what is the innate immunity that is present, environment is present at the time that is immunization occurs. And that is why we feel that is very important in the IEDB to capture not only the epitope, but also what is the context, what was the experiment in which that biological activity was noted. And again, is there any IEDB alternative for innate immune system, from Omar Shaikh? And to the best of my knowledge, there is no database that records structures related to innate immunity.
Okay, I have two related questions, probably the same user: how to select multiple alleles. We can only select 20 or only 15 alleles. I am not aware of that. Tomorrow we are going to go through in detail for selection of alleles, and we can test it. If that's not clear, then please answer the question again. Then Rizwan Ahmed asked, is it possible to compare a particular epitope simultaneously in different diseases to check if there are overlapping possibility? That's a very nice application. So in these cases, we would recommend to do two separate queries, one for each disease, download the data, and then either, if you want to have exact matches, I mean, use Excel or whatever, but you can also use the clustering tool to identify in which cases do you have overlap between two lists of peptides. And the clustering tool we're going to also talk about more later. Then Alexei Parolo talks about BetPrepared 3.0 and asked, are we going to implement it? I mentioned this before. Hopefully that's in the answered list. Otherwise, we can also talk about that tomorrow. And how to suggest other tools for B-cell epitope predictions. Please use the help system. Just email us. We are very curious always. There might be reasons why we don't implement something such as licensing or also impact on server resources. So not always can we have everything. And actually the most common thing is that there's plenty of papers out describing methods that are actually impossible to re-implement, and then nobody has the code even available anymore. Okay, that's it for me, Alex.
Not sure.
Awesome. And I think I'll give you one more minute to take a look because we are at time. So Alex and Bjoern review the last few questions. And as they do, apologies in advance if we cannot get to all the questions. We will do our best, of course. If not, we can try and type the answers as well. But as Bjoern mentioned, if there's anything that we don't get to, you can email us at help@iedb.org. Also in the feedback survey, there is a section for you to provide just general comments or suggestions. So we will not miss any of that. But anything else, Alex or Bjoern, from yourselves?
No, I just wanted to stress, I think many of the questions that I have here still open are going to be much better tomorrow, where I don't have to rush through everything. And I'll try as much as possible to actually cover these points as we're going to go through the detailed version of the tools. And I'll try to answer some more in the chat, but I also have to, before we close that out. Yeah, but I think we should stop here.
Awesome. So a massive thank you to Alex and Bjoern, our wonderful principal investigators of the IEDB. Thank you both.